Perfect. All right. So, okay, yeah. So, thanks a lot for this great introduction. And yeah, I just mentioned the title of the talk, Decoding Life. So, let's get into it. So, the deoxyribonucleic acid, right? So, this is a molecule that encodes what defines every single living being that lives on Earth. And then it has been passed on from generation to generation as an Olympic torch since billions of years ago. However, this manual did not come with its own manual. So we had to, so everything we know about this molecule today is a product of decades of work from several researchers that developed brilliant ways in order to understand exactly what this molecule is. So the presentation that you're seeing right now is basically a story of how we came from knowing absolutely nothing to all the knowledge that we gathered right now. And basically, this talk is divided in three parts. So at the beginning, we're going to look at the early biochemistry experiments that allowed us to understand what DNA is made of. Then in the middle, we're going to talk about some state-of-the-art techniques that are called high throughput because they allow us to sequence high amounts of DNA in a short period of time. And then at the end, I'll talk about some applications and perspectives that we have now with these technologies in hand. Right? So buckle up because we're going back to the 19th century in 1989, where a Swiss scientist called Friedrich Michel, um, he had a deal with a hospital close to his lab, where he was able to get some bandages from patients. And these bandages were they were embedded with pus. And his goal in taking this pus was to understand what would be the proteins that constitute the immune cells that help our body protect itself from external threats. While studying the composition of this pus, he found out that actually uh, immune cells, he found an interesting substance which was not exactly a protein. And then this protein was made of basically hydrogen, oxygen, nitrogen, carbon, and a high amount of phosphorus, like higher amount than anything else that we have in our body. And then this substance got him intrigued. So as he started narrowing down his experiments, he found out that this substance came from the nucleus of cells, and therefore he called it the nucleus. Uh, all right. So in parallel to his, okay. So before I go there, so during this talk, I have 5 links that will be referred to that will be in the description of this video when it goes up to YouTube. And then the first video is actually an interesting procedure that is published in it's a YouTube video as well in the channel Nile Red, where the author he shows that we can nowadays with things that you can buy in stores you can extract the DNA out of fruits and vegetables, and you can do that at your home if you'd like to. So, pretty interesting reference. So, in parallel to Misha's work, Walter Fleming, now a German scientist, he started staining salamander cells with aniline. And then it was interesting because in the nucleus of the cells, he found some substance that could also acquire all this aniline and get like really easy to see in the microscope. So actually here you can see the drawings that he made while observing the cells in the microscope. Due to this property of absorbing all the color that he added to his substance, he then named these things the color bodies or chromosomes. And then one of the very interesting properties of these chromosomes is that as cells divided, you also see that the daughter cells, they had chromosomes just as the mother cell. So this was already the principle of observations of something that could explain the heredity that we see, for example, like a human having a human as a kid or a plant having coming from a seed that also came from another plant, right? So 20 years later, so right now at the beginning of the 20th century, we have Walter Sutton, which is an American scientist. And then him was also using the same aniline techniques, but then right now he was counting the chromosomes that he could see in the microscope. So by using a study with grasshoppers, he was able to count that in the sperm and the eggs, you have half of the number of chromosomes that you have in the other cells of your individual. And then during the fecundation, these two quantities, they join each other. And then you have the embryo that will then make a new grasshopper. So at this point, he had access to the manuscripts of Mendel with the piece that were forgotten for quite a while. And then he was able to see that these chromosomes, they would be the physical basis that would show how, for example, peas, they could share the material, the genetic material in half, and then pass on to the next generation, which would then join together and then explain, for example, the relationship of dominant and recessive genes. So going back to the biochemistry of DNA, I will bring up two researchers. So Albert Cossel, which is also a German scientist, and Phoebus Levine. So they did not live around the same time, but Cossel and Levine, they were great biochemists, not only for DNA, but also for several other biological substances. And Levine, at the beginning of his career, he visited the lab of Cossel. So basically, Cossel was able to characterize the DNA with the building blocks that Misha had identified before, was composed basically of phosphate, sugar, and nucleotides. So the phosphate was the form in which the abnormal quantity of phosphorus is trapped in the body, then composes the nuclein and the chromosomes. And then the nucleotides are already the four entities that we now represent with the famous letters A, T, C, and G, which you can see here in the screen. But then throughout this presentation, I used the work of one of our amazing illustrators here at Kytos, Loma Branco, because, like, they're so cute, why not? All right, so then Levine, he knew about the composition of the DNA at the molecular level, and then his idea, and then the challenge that he decided to take on was, okay, so how we can organize the sugar, the phosphate, and the nucleotides into this structure that is the DNA, right? So at the beginning, he thought that all these nucleotides, they would be together to form the basic unit of DNA, and he proposed several structures, but actually he never managed to get evidence enough in order to show that the DNA is made of these four nucleotides together. And then the current that he developed throughout his career is today known as the tetranucleotide hypothesis. Okay, so right now we're going to go to 1952, where Irving Chargaff, which is also, so right now he's an American scientist. He looked at the different proportions of these nucleotides in different species of sea urchins. And then something that he found that was very interesting was that the ratios of nucleotides can change in different species. However, if you look at the same species, the ratios of nucleotides are the same. So here is already showing that the composition of nucleotides can be what defines what a species is, and also what differentiates one species from another. Something that was very interesting as well is that he saw that the proportions of adenine and thymine were very similar for most of these species, for all the species that he analyzed, actually. And also the proportion of guanine is very similar to the proportion of cytosine. Cool. So the next breakthrough in the structure of DNA came, actually, it came around the same time when a scientist called Rosalind Franklin, which is also being represented here in one of the cards from the game Replica that Mariana mentioned at the beginning of the video. So Rosalind Franklin, this scientist that I decided to homage, she was trying to crystallize DNA using different proportions of substance. For example, different amounts of water, different amounts of salt. And then as she was optimizing her procedure, she then would crystallize the DNA and then see which patterns of diffraction of light, of X-ray, these crystals would cause. And therefore, she created this picture, which is commonly known as picture 51, which was the basis for the discovery of the structure of the DNA itself. And if you'd like to understand exactly how this image here allows us to understand the structure of the DNA that we'll show really soon, you can check for the second reference link that will be in the description. So over here, you have another YouTuber that uses a setup with lasers and lamps of tungsten to show how the light can be diffracted when it goes up something that is helicoidal. And then, well, as I mentioned, as I just mentioned, DNA has this helicoidal structure, therefore produces the reflection pattern that you just saw. And then the structure, and then basically the structure that we see today was made like in the 50s as well, where we have a double helix of phosphorus, the molecule that was already very important since the beginning of the DNA story. And then these two helix of phosphorus in the form of phosphate, they're linked by the base pairs, by the nucleotides that have also been identified. And then these nucleotides, they are organized in pairs, as we have seen. They're organized in pairs. And one interesting point that I would like to show here is that these pairs of nucleotides, they contain one purine, which, if you look at the structure, are molecules that contain two cycles of carbons, with a pyrimidine, which has only one cycle of carbons. And then it's quite interesting because you have like a molecule with one cycle of carbons and a molecule with two cycles of carbons. And then if you have this kind of matching, it's very easy for a molecule to see, for example, if you have a pairing that is not good, for example, two molecules that have only one cycle, and then they're going to be further apart from each other, or two molecules that have the two cycles as well, because then you're not going to have enough space in your double helix. So this allows us, the cellular system, to understand if something is going on wrong very easily. And then, well, if you think about it, this system does not prevent an adenine being paired with a cytosine. And this is why the hydrogen bonds that link these two molecules, they're very important, because an adenine and a thymine, they link with only two hydrogen bonds, while guanine and cytosine, they link with three hydrogen bonds. So then you have this other layer of information, which then shows the number of hydrogen bonds. So the molecular machinery can understand as well if you have like a thymine paired with a guanosine, for example, which is also not good for cells. And then the person who look at the pictures generated by Rosalind Franklin, it's Watson and then Crick. And then Crick is also being homaged in our game, replica game. And then besides the molecular constitution of things, the double helix is also very interesting structure. It's a very interesting structure because it allows us to do reproduction by just splitting each one of the helix apart and then completing the pattern in each one of them, like you can see here. So basically you have one molecule of the DNA, and then from this one you can have two other molecules of DNA, which contain one helix from its parent molecule and another helix, here represented by the black one, which was just added based on the template that came from its parent. And then on the other side, if for some reason you have a damage that is occurs to your molecule, you can also have other enzymes that can just come and complete again your DNA with the same sequences that you had before without loss of information. And the enzyme that does it is called DNA polymerase. And then the DNA polymerase was discovered by Arthur Kornberg in 1971. And then here, I will recommend you to the third link in the description. So basically, this link was made by the channel Wahi Movies, where they show in very nice detail exactly how the DNA replication can happen at the molecular level. And then they also show several other biological processes that are very interesting. All right, so by getting to understand the structure of the DNA and the molecule that allow us to both replicate it or repair it, we have several doors open. And then this was just humanity passing on to another level, So at this time, there were several techniques that were developed, and those techniques are still relevant today, even though it's been 50 years. So one of them, as you can see here, and you've probably heard this acronym recently, is the polymerase chain reaction. So the idea of the polymerase chain reaction is that you can take one sample of the DNA, and then you can amplify this sample to higher levels where it will be way easier for you to detect what exactly this thing is, right? So then how does the PCR works? So you will start with a molecule of DNA, and then if you warm this molecule of DNA to the range of a little bit more than 90 degrees Celsius, this molecule will denaturate and then will separate itself in two parts. If you cool down your sample to around 50, 60 degrees in a preparation that contains both the DNA polymerase and several nucleotides isolated together. Sorry, So if you put this down, first of all, you need to add some structure that we call primers, and then the primers they will allow the DNA polymerase to bind to the sequence, and then as you cool it down, the DNA polymerase will then use these primers in order to bind to the sequence, and then it will complete the whole sequence with the substance that you have in your cell. And then you can go from one step with one molecule to two molecules. But then if you just repeat this same procedure over and over, you can keep amplifying your sample. So right now, by having two fragments from your first fragment of DNA, you can just warm up your sample again. You're going to denaturate these two sequences. Then you have the primers that will bind it to the beginning of those sequences. And then as you cool it down, the polymerase will then use all the nucleotides that are available in the solution to then create another strand of DNA. And then like this, you can start amplifying your DNA indefinitely, like as much as you want. And this technique, which we still use to diagnose diseases, was also very important for the next step. So the next step is thinking about what we are amplifying, right? Because so far, we have the DNA, we know what it's made of from the atom level, we know the molecules, we know how they're organized, but then we don't know the sequence of how these molecules they are put together in the DNA, right? So in order to solve this, to answer this question, so I have a scientist called Sanger, which developed a model technique that allow us to see the sequence of the fragment that we're looking at. So here we have our DNA sample, which then we can, it already has the primer here at the beginning. And then in the same sequencing idea is that we will again use the same enzyme, the DNA polymerase, with a pool of nucleotides. But in this pool of nucleotides, we're going to choose one, for example, T, and then in this, we're going to add some T molecules, like thymine, that have a part of the molecule missing. So namely, it's like one of the group OH, the hydroxyl ones. But basically, the fact that you have this nucleotide that is not complete will allow the polymerase to keep building your fragment of DNA. But then when you have this molecule, it won't be able to continue anymore. So if you do this type of amplification, you end up with very short fragments where the T got in the sequence at the beginning. You also get some medium fragments where you had more molecules being added to a fragment before this deficient nucleotide came. And then you will also be able to construct some fragments that will go all the way, and then you can have the whole sequence of your fragment. So you're going to repeat this procedure several times in your, you're going to repeat the PCR in your sample several times, and then you're going to end up with this mixture that has incomplete fragments of DNA in different parts all the way to your whole fragment. So we did this for letter T, and then you can do, so we're doing this for letter T. So we have our samples, we put them all, we put them all together, but then we're going to put them at the beginning of a gel, and then at the extremities of this gel, we will apply electric field. So here, what will happen is that since DNA has a has a charge as well, it will move throughout this gel. But the shorter fragments, they will move way faster than the longer fragments, because the shorter fragments, they are lighter and have less resistance from the gel. So as you do this whole procedure with your samples, you're going to get the gel. And then at the different positions in the gel, you will have different fragments with different lengths, right? So we're going to have the shorter fragments all the way at the bottom, and then we'll have the longer fragments all the way at the top. So if you repeat this for each one of the bases, you can get some preparation that gets something like this. And then the cool thing here is that by looking at the places where you can see your molecules, so basically these black blobs, they are exactly these groups of DNAs over here. So yeah, you can see exactly where each letter is in your fragment. So you come from something that you have no idea what it is, and then you come to this structure here. So for this example, we have this sequence that we start from the bottom. So we have like a T at the beginning, then an A, and then C, G, A, G, A, T, A, T, A, T, G, G, C, right? So this is a kind of rudimentary technique, but it was the first time that we could actually look at the molecule of DNA and say, okay, this is this sequence in which the nucleotides are being added together. And if you'd like to check the original gel from the first publication that developed this technique, you can follow this link and then look for the playthrough. Great. So by knowing how we can sequence DNA, then the biggest question was then, what is a human DNA, right? So because yeah, we know that this is the basis of life. We know we're made of it, but then yeah, like uh, how a human can be described in terms of DNA. So around like 10 years after the technique has been established, people started discussing this at a serious institutional and political level. And then in 1991, the US government started what is known as the Human Genome Project. And then the first goal of this project was not to sequence the whole DNA, but was to get some landmarks to get the point, the kickstart, the point of start for the determination of the whole sequence of the DNA. And actually, in 1994, the consortium that was working in this project was able to publish a map of the human DNA with certain sequences that were roughly 1 million base pairs apart. And then people already knew that our DNA had 3 billion base pairs. Great. So, still in the context of this project, 2 years later, the scientists that were working in order to understand what is the sequence of the human genome, they met in Bermuda, and then they published what is today known as the Bermuda Principles, which were basically guidelines for people to publish their research as early as possible, so labs could collaborate on an international level in order to tackle this challenge, right? Because sequencing 3 billion base pairs, it was a challenge that was huge. Right. So then in 1999, this consortium was able to publish the first chromosome. So the first chromosome to be sequenced was the chromosome 22, and then it was not chosen by hazard. You know, if you look at the chromosomes, they are actually ordered by size. So the chromosome 1 is the biggest one. And then the chromosome 22, apart from the sex chromosomes, was the smaller than. So this was the first chapter of a long journey. And so the chromosome 22. But then, 2 years later, the International Human Genome Sequencing Consortium was able to publish already a draft of what would be the whole genome, right? So it's quite interesting because from 94 to 99 was the time that it took to sequence the first chromosome. And then from 99 to 2001, we were able to sequence all the other 22, right? Or the other 45. And then in 2004, they published the final version of the first genome, because we had done others, like we had updated this genome before. But then the first version was published in 2004. And this was like a great achievement for humanity, because we could finally understand what is this DNA sequence that makes a human, right? So if you look at the paper, they have several interesting statistics. So for example, so here was the time that we knew that a human is made of 20,000 genes. Great. So if you think about it, like if you think about this project, we had lots of people that were working in these techniques, and then there were lots of people that are also optimizing the different techniques that we could use in order to sequence DNA. So right now, I'm going to show you the last technical part, but then this is how we sequence DNA today, right? So basically, this picture that you can see here is what we call the flow cell. And then in this flow cell, we have 4 different lanes. So the sequencing happens in each one of these lanes. And then I will show you exactly what happens here. So these lanes, they are already coded with a little sequence of DNA. And then this DNA, it has the same sequence here. We will then put our sample in each one in this lane over here. And then when you put our sample here, we will have our fragments, our DNA fragments. But then in order for the fragments to be able to interact with the coating, we had to add a sequence at the end of each one of the fragments during the preparation of our DNA, which we call adapters. So basically, you get your DNA fragment and an adapter, what is usually termed as a library. So if you read about DNA sequence on the internet and then you find a library, so library is basically your fragment treated already in order to be able to be sequenced. So, okay, you have this little piece of glass with your DNA that is attached to the bottom. And then what you do is that you will inject a solution. Yeah, and then you can see here. So basically here will be the vision that the machine that contains the flow cell inside of it has over here. So basically you're going to wash this little lane over here with a solution containing, for example, adenine, so our nucleotides as always. And then this is a version of adenine that is also attached to a fluorescent probe. So basically, when this adenine, which is here in the medium, finds, for example, AT, it will bind to it, and then it will glow in the flow cell. So basically, you're just going to take a snapshot of what happens when you wash it with adenine, right? So then you repeat the procedure, but then now we're going to add another nucleotide, for example, T. And then as T comes to the fragments that contains its own molecule, you will have another dot corresponding to the color of the probe that you attach to T. Then you can just repeat it for all the nucleotides. So we have G, and then C, and then C find its position here. And then you will repeat this procedure several times, like A, and then T, and G, and C, and so on, until you have all your fragments. Like actually you repeat it as many times as you want. So here repeat 5 times. And then, sometimes you can have fragments that are larger than 5. But then, so this is one of the parameters for analysis. And actually it can be more expensive if you ask for the company to do longer reads. But all right. So if you think about the whole procedure, what you get in the memory of the machine is basically a series of photos where fragments at the same position, they will shine with different colors at different times. And then basically this thing here, you can later look at the color of each, you can look at the position of each fragment, and then you can see which colors had been shown up at each time. And then from this, you can decode the sequence of the DNA that you had. And then this is quite an interesting thing, because right now we can put millions of fragments inside one single lane, and then you can just take pictures over and over. And this is a very interesting technique that allows us to go really far. So the thing is that when you finish your analysis, you're gonna get lots of fragments. But then the question is, how can you turn these fragments into genome, right? So basically, during the same processes of during the same biochemical evolutions, we also had some evolutions on the computer science side of things, in which basically, if you look at the colors or the sequences at the border of your fragments, it allows you to kind of pile them up and then find, for example, a fragment that will have a sequence from both the first and the second together, in order for you to know, okay, so now we have a longer fragment that is a longer fragment that allow us to keep extending, and then like this, go from several little fragments to a big sequence of nucleotides. On the other side as well, if you already have the genome of the organism you're studying at, now you can do the same type of procedure, and then you can know which parts of your genome will have higher or lower affinity in the preparation that you had at the beginning. So right now, I'm just gonna show, for example, like another technique that is still not widely used, but looks very promising and very interesting from the point of sequencing. And also, it allows us to sequence longer fragments. So then it's also very good for us to discover the genome of other species, for example, with with yeah, with more accuracy. So basically, what this technique does, the name of this technique is called nanopore. So basically, I have this gray membrane with a pore that is basically a protein, like this green protein over here. And then if I don't have anything inside this pore, you have a flow of ions coming from one side of the membrane to the other. But then since ions, they have electric charge, you pretty much have a electric current going through this pore. And with this, you can measure what is the difference of potential, electric potential in the two sides of your membrane, and then you're going to get this signal over here. And then the thing is, as you allow your DNA to pass through this pore, and there are specialized proteins that will do that for you, and they're still being optimized nowadays. Depending on the sequence that you have here, you have a different profile of electric signal. So then by looking at the evolution of the electric signal over here, you can sequence pretty much a fragment of DNA, and then it's cool because with this technique, if you have the proper conditions, you can pretty much sequence a whole chromosome right away. All right. So I just told you about like all the hassle that people have been going through in order to understand what are the chemical components of DNA, all the computational challenges that we had in order to take this biochemical uh, the biochemical improvements into turn this into a sequence that is 3 billion letters long. But then, like, what do we do with it, right? It's very important as well to think that, yeah, knowing about the human genome, it's a very nice from a philosophical point of view. But then, why should society bother with this kind of thing? So the first thing is that it allows us to improve the health, the life of people. So for example, the first thing that we can do once you have the ability to check for the sequence of DNA from patients or even tumors is to compare the sequence that you have with a healthy individual, and then you can see if you can find, for example, a mutation. And then this can show that you have a patient that either has a genetic disease, and then this genetic disease is just due to a change in DNA sequence that then makes a protein that is faulty, or you can also find a tumor that then also has a mutation specifically in a protein, and then if you can somehow manage to develop a drug that can help you to deal with the lack of this protein, this can also reduce the symptoms and all the like the problems associated with a disease or even to cure it. Right? On the other side, if you also look at different mutations or different variations that people can have, depending on where they came from in the world, you also have you can also start attributing the places where these people probably came from, because I have lots of people that came from this region that have the same type of variants in their own DNA. And then this is the basis for what is very popular since recently, the ancestry tests. And then on the other side as well, the some of these analyses over here, they can also help you understand if your DNA could make you more prone to develop certain types of diseases later in your life, which is a very interesting thing. However, I would also take this kind of information with a grain of salt. And then there's this very interesting movie called Gattaca from 1997 that discuss as well the advantages and the problems of using this kind of analysis to base very important decisions in life, like which career should someone take or how much this person should pay for their health insurance. On the other side, we can also use this type of analysis also to in the field of forensics. So basically, if you have a crime scene and then you find some blood samples or hair or even skin, You can also take these samples and then you can amplify the DNA over there, and then you can use this DNA to match with possible suspects that have been at this crime scene. And then with this, you can characterize with high confidence who would be the person that was responsible for a crime. Even though, excuse me, there's also the fourth reference for this video, which talked up as well about a person that committed several crimes, and this person, he never really he never really got caught, right? Until the police department decided to take the DNA samples that were found in every single one of these places, because like they knew that was the same DNA, and run it with some databases of people that performed ancestry tests. And then they did not find the suspect itself over there, but then they found close relatives. And then this information allowed people to turn down who was the murderer, and then actually to make justice. But then we also know that this kind of technique can also raise some privacy concerns, right? So yeah, so then in the link 4, there's a video from the channel Veritas Room in which he discussed the case, which is the Golden State murder. And then it's a very interesting discussion that I recommend you to follow up through if you're interested. On the other side, by being able to gather the DNA as well in the case of natural disasters, allow us to recognize the victims and also to help their family to cope with the loss. And then finally, there's also this field that is developing quite fast now, and it's called metagenomics. So basically now here, you're going to take one sample, and then we're going to sequence everything that you have in there. And then the metagenomics has special types of techniques that allow you to see different species that could be living at the same place. And then this is a field that has lots of applications, for example, in agriculture. So to understand the type of soil and this type of interaction that plants could have with the soil, so then you can improve the production of food. We can also understand how animals can interact with each other in a global ecological system. We can also use metagenomics in order to find, for example, bacteria that probably have an enzyme that we could use in order to treat a disease as well. And also we can take samples from water treatment centers, and then we can use metagenomics in order to see which type of organisms could be living in this water, and and then assess the quality of this water. And it might look a joke, but actually, if you take also the samples of our intestines, we have a rich ecosystem over there with several bacteria. And then it has been shown already that the bacteria that live in our intestines, they're associated to several processes in our body, so they can increase or reduce our predisposition to certain diseases. They can even modulate our appetite. So for example, like if you eat lots of greasy food, you're gonna nurture the bacteria that actually live off grease, and then these bacteria can then release some chemicals that then will go to your brain and then make you crave for more greasy food. And also then you have this cycle in which the bacteria is kind of manipulating you to eat more, right? And then if you'd like to know more about it, so then we go to the 5th and last reference that I mentioned. It's a video from the channel called Kurzgesagt, and then they talk about how the microbiome develops a very important role in our lives. So after this big journey, let's wrap up everything up. So what I would like you to remember at the end of this talk is that basically the final structure of the DNA was a result of several decades of work from lots of people that worked pretty much on the results that were produced by the previous generation. Also, for us to be able to construct the human genome itself, like we needed a international collaboration effort. But actually, this international collaboration was very efficient and hand out really good results in a very short amount of time. In one point, it's also important to think that even though these technologies, they are great for us, they can also be used for the bad. And then like it's up to us as a society to understand what we want and how we will use these tools. And also, like, we're still learning. So even though the journey from the beginning up to here can be seen like a long one, there's still way much more that is still possible, and that, yeah, we can do amazing things with it. So I'd like to thank you for your attention, and if you have anything making you scratch your head, I would like to talk about it right now.